It's my pleasure to welcome everyone to the next instalment of the MPCCC Precision Oncology seminar series. My name is Mark Shackleton. I'm the clinical lead of the MPCCC's Precision Oncology program, and in my spare time, the Director of Oncology at the Alfred Hospital. I'd like to open today's seminar by acknowledging the traditional owners of the lands on which we are joined together today, and respectfully acknowledge their elders, past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who are present today. The MPCCC Precision Oncology program essentially represents a community of scientific researchers, health professionals, and academics across Victoria who are interested generally in precision medicine, and specifically how it applies to patients with or at risk of cancer. The program's working to establish better access to molecular sequencing for cancer patients particularly across the southeast regions of Victoria. But increasingly, and I think increasingly importantly, we also support and host several tumour-specific molecular tumour boards to support clinicians in the interpretation and clinical application of molecular data, particularly as it pertains to linking to therapies either available via clinical trials or other programs. These seminars are another important part of the Precision Oncology program, and they certainly help us to share and catch up with the latest research activity across our own partnership, but also in Victoria, and in fact even internationally. We've been fortunate to welcome international speakers to present at this forum, which has been fantastic. Today is a real honour. I'm so pleased to welcome Professor Sean Grimmond, the Director of the University of Melbourne Centre for Cancer Research, where he leads the Cancers of Low Survival and Unmet Need initiative that we'll be talking about today. Amongst numerous honours and also responsibilities, Sean is also the co-chair of the VCCC's Precision Oncology program. Sean is one of the real rock stars, not just nationally, but in fact internationally, in the field of cancer genetics. He holds a PhD in the area from the University of Queensland. He's a scientific fellow of the RCPA and a fellow of the Australian Academy of Health and Medical Sciences, and his research has really been quite transformative, applying groundbreaking genomic technologies as they've rapidly emerged and come onto the scene to better understand mammalian biology generally, but particularly cancer. Notably, for the last 10 years or so, he's led Australia's International Cancer Genome Consortium efforts, so the ICGC, which have pioneered large-scale whole genome and transcriptome sequencing to identify mutations and potential therapeutic vulnerabilities in some of the most difficult to treat cancers. Currently, he's focused on clinical genomic analysis of cancers, particularly, again, clinically challenging cancers, and also ex vivo drug screening of cancer cancers using various laboratory modelling processes. Sean's talk today is called the Column Initiative, using comprehensive whole genome and transcriptome analysis to improve diagnosis and therapeutic selection for cancers of low survival and unmet need. There will be some time for questions at the end of the presentation. And please turn off put your computers onto mute during Sean's talk. Without any further ado, I will hand over to Sean. Thanks so much, Sean. Great to see you. Looking forward to your talk. You've been very good, Sean. You put yourself on mute. I'm very impressed, but you need to take yourself off mute if you're going to actually talk.
I have already. I was actually, that bit there, I was just thanking you for such a warm introduction. It's not very often I get called a rock star these days. But if I need an agent, I reckon you're the man for the job. Look, it's an absolute pleasure to be presenting here today. What I wanted to do with today's talk is really describe to you a large multi-lab initiative coming out of the University of Melbourne Centre for Cancer Research that has become the engine room for a lot of translational and clinical studies now underway across Melbourne and a number of collaborating groups. It provides a core set of rapid comprehensive genomic testing capability to the VCCC Alliance, but also a number of other collaborating programs. And what I really wanted to do with today's talk, I guess, is give you an overview of how we've come about to undertake these very comprehensive whole genome transcriptome approaches in some cancers, show how we're targeting cancers of low survival and unmet need, and I guess really trying to show you where the utility is we're already seeing from those current efforts is appearing. I guess also I want to try to describe to you how we've been working through as a group here within the VCCC. How do we make sense of molecular testing? How do we feel and find our way to work out when is it appropriate to use a panel test, when may be appropriate to use a comprehensive test, and where do we think the benefit might lie in the use of some of these approaches? I apologise. A minor code yellow warning here within the building. So, all right, just by introduction, we have had a long-standing interest both within my laboratory and indeed within the UMCCR to focus on cancers of low survival and unmet need. And I guess this is really a slide that has been in many of my talks and is a core motivation to our work, and relates to, in this case, representing the changes in 5-year survival for a selected set of cancers. This is Australian data since the 1980s, showing you that overall we've been seeing a steady improvement in survival. And while we've had some great successes, when we look in the bottom right-hand corner, we can see that there have been plenty of cancers that have been left behind up in that top left-hand corner. There's a variety of reasons for this. Some of these cancers are just inherently recalcitrant, like pancreatic cancer. Some of these are particularly rare. Some of these are a diagnostic conundrum, so it's difficult to actually treat a cancer when you use traditional approaches where you don't know where it comes from. That's an example of the unknown primary. And some of these are quite aggressive, so we haven't got the opportunity of trialing lots of different drugs to come up with something, even in a traditional trial setting, that might work on those patients. So we've been very keen to see how we can use molecular approaches to decode these genomes for these particular cancers, and in a research setting, try to come up with an atlas of what we believe may be the root causes of those malignancies, and where may the druggable targets or insights come from that genomic sequencing that might be used to improve the therapy in some of those cancers that we're really seeing in that top left-hand corner. So the way that we've gone about doing this is another common slide in my talks, is that we very much see, if we want to see the likes of precision oncology be realised, there's a number of steps we have to walk through along the way. And the first one really is building up a foundational knowledge base of what is driving cancers one patient at a time, and then see how we can map that to our actionable segments, be able to then use that robustly in a pathology environment so that we can turn it around in a clinically useful time frame, and then use that to drive the trials and bring in new therapies that will see the improved outcomes. As Mark said, much, or in fact all of the work that we really do is based and stood on the shoulders of our colleagues within the International Cancer Genome Consortium, which includes the US's activities in TCGA. And this has been a huge international effort, which has been systematically building better and better atlases for us to use as references for our work going forward. And really the way those studies tend to work is we are steadily accruing patients. We put those into cohorts. We then will run a number of analyses trying to find the genes that may be likely to drive that particular disease. Here I'm just showing you the classical sort of first steps we did for pancreatic adenocarcinoma, looking at about 450 patients. And we end up seeing there's a large number of genes here in this plot, some of which are being mutated very frequently, like KRAS, and then a series of others that get less and less frequent. But that rather chaotic, complicated picture we can see there can be redrawn a second way, and that is if we start to take those genes that have been mutated and look at what pathways or processes they fall into, we actually find that in pancreatic cancer, the driver genes really coalesce around a core set of pathways. And this sort of process of identifying the drivers, identifying the processes, has been now carried out in most major cancers and in an increasing number of rare cancers. But it really does give us that blueprint of what do we think might be underlying a particular malignancy. The colours on the right-hand side are just to remind me that this is where, once we found these genes to be drivers through the traditional discovery approaches, there's validation or orthogonal tests that have been done to show that they are bona fide involved in promoting or allowing this disease to thrive. I guess crucially for us is if you actually have that set of genes, and from a biological point of view, that's very useful. But if you're a patient with one of these malignancies, what you want to know is, are any of these driving mutations that might occur in your tumour potentially actionable? And in fact, even though we know that pancreatic cancer is something that is notoriously hard to treat, if we look at the underlying genetics and then look at what are the potential therapeutic options that could be considered about some of those mutations, there is actually a wealth of them. But the challenge for us is that the relative frequency of these types of opportunities occur at low frequency. And because of the late stage and the aggressive nature of this disease, if you really want to be able to exploit these sorts of vulnerabilities when they occur in very small segments of your overall cancer community, what you need to be doing is coming up with an approach where you can rapidly make sense of their genomes or the sequences, and then be able to assign those ideally to more targeted approaches to actually test whether there's going to be benefit coming from them. So I guess really this is something that has been going on steadily for the last 10 to 12 years, where people have been looking at each particular cancer. In many cases, it's been done by exome sequencing. ICGC is certainly focused more on whole genome sequencing. And we've been able to understand the root causes in individual tumours, and we will come up with an understanding of that particular cancer type. But because of the nature of the way the studies were undertaken, they'd never really been integrated into any sort of way to give us a truly unified playbook for creating the foundations for molecular pathology for both solid and hematological malignancies. And indeed, there has been a study which is really focused primarily on solid malignancies just recently. It's called the Pan-Cancer Analysis Whole Genomes. And this is an effort whereby we've carried out a completely unified, harmonized analysis of 38 different cancer types, whereby we've systematically tried to map out what we think is going to be the crucial information about what causes those cancers to develop, what is the commonalities between cancers, and what are the types of events that can ultimately drive those cancers through their progression. All right, so just to highlight a couple of take-homes from this, because it really does, you'll see it'll become crucially important when we want to start to apply genomic testing to different cancer types going forward. This is a plot to show you the way that PCORG, or the Pan-Cancer Analysis of Whole Genomes, now tends to view somatic damage across cancer types. They've been able to create these enormous sorts of tables whereby we have genes as columns, cancer types as columns, and cancer genes as rows. We're able to determine the relative abundance or frequency at which those cancer genes are being damaged across each cancer type. And then the panel on the far right-hand side is to show you that when it comes to the nature of that damage, it can come in many different types. It can occur in coding sequences, it can occur in non-coding sequences, it can occur in regulatory sequences, it can occur in non-coding sequences and lead to rearrangements of those genes. So there's quite a complex range of damage, and for different genes, we can see different propensities and different types of damage to those. That will become important later on when we think about what's the assay that we want to use for those particular events. Indeed, if you take an overall global view of the PCORG data and you have a look at how many events do we see on average as being a bona fide driver mutation within a malignancy. On average, it's about 4.5 or a little over, so between 4 to 5 driving mutations per malignancy, and indeed 2 to 3 of those are coding mutations, and an additional 2 to 3 are non-coding. So this is once again another hint that while panel-based sequencing or exomes can be great for coding sequences, we may not be getting the whole story. The second thing I would point out here is that in terms of a germline contribution, in this particular study, none of our cases have been collected from familial cancer families. They've all just been collected as a presumption of being somatic cases by and by. So the frequency of 17% of our patients having a truncating germline mutation is clearly a conservative underestimate of the level of germline contribution that we're seeing across the board. So another key consideration when we start to think about working in a testing setting going forward. I guess if we have a look at our scorecard now, based on where we got to with PCORG, we can now see that for the majority of cases, it's over 90% now, and indeed in many cancer types, it's well over 95%. We're able to detect driving mutations in those malignancies. There are some cancers at which we're still clearly missing some of the events that are driving those disease, or we can find no chromosomal genomic event that's driving those. So you can see that some of the renal cell carcinomas and indeed some of the CNS tumors, there's an underrepresentation of drivers, but by and by now we're able to get quite a complete profile for the majority of cases. A final thing that you'll see becomes a very powerful tool for us later on is this realization that when you accumulate damage, either through a mutational exposure or a defect in DNA damage repair, for instance, this leads to telltale damage across your genome. That type of damage, if you just sum up globally across the entire genome of a particular malignancy, can be analyzed mathematically to work out what is the relative contribution of that somatic damage to a pre-existing mutagenic process. And this is known as mutational signature analysis. And what it's really allowing us to do is a number of things. The first thing is we're able to take a sample from the ICGC set, determine based on these somatic scarring patterns that are discrete for different processes. We're able to look for the presence or absence of them, and we're able to quantify them. And the graph or the images I'm showing you here are just showing you the relative frequency of trinucleotide sequences that have a very distinct pattern across a set of 96 trinucleotides, for which these patterns are now known to occur in the tumor somatically in the presence of HR, or in this case, BRCA pathway mutations, or indeed, in this case, we're seeing a very discrete pattern that is related to poly deficiency, Pol D deficiency, mismatch repair, the list goes on and on, base excision repair, indeed, base excision repair. These events can arise either from being a germline feature that's been inherited, or they can arise as a somatic event within that patient, or indeed, if you see epigenetic silencing of some of these pathways, these telltale signatures will appear. This gives us a very powerful tool for orthogonal testing of our genomes to actually go back and look at the genetics that we see when we're trying to understand what genes might be either driving those or being the root cause of the mutation. Should also say that we can quite readily now detect the exposure to a number of mutagens that are readily identified within the PCORG set, whether this be the carcinogen from tobacco, whether it be UV exposure from the sun or other agents. We've now, once again, you can see these telltale signatures that can be identified, some of which clearly will have some benefits when it comes to understanding tissue of origin, for instance, in an ambiguous tumor. And then finally, we know that there are some now exposures to pathogens that can give rise to very specific signatures quite important for GI cancers. And then finally, of course, we've got an increasing number of signatures that we see in patients that have undergone multiple lines of treatment, where we can actually see the consequence of some of the chemotherapies, for instance, that we give to our patients. The final thing that's really come out of the PCORG studies, which we've now taken from that discovery phase and are integrating into our pipeline, is this idea of using the background passenger mutation rate across the genome to infer tissue of origin. And colleagues within ICGC, led by Lincoln Stein, carried out an effort whereby they took a number of genomic features, background passenger mutation rate being the major one, and then tried to see if you could look at that background passenger mutation rate across the genome to create a machine learning tool, which could then go through and predict tissue of origin in subsequent tumors. And this approach now has been worked to a point that we were seeing in the order of 90% accuracy with that caller. And I think this really opens up some interesting opportunities when it comes to cancers with ambiguous diagnoses, or indeed just quality checking some of the more difficult tumors sometimes people may be dealing with. And this is really something that we can get extracted straight from the standard genomic testing of individuals. Yeah, I'll leave that there. All right, so if that's our foundations, that's what we've really been working on. And then we've been looking to create an engine that we can use that then in a clinical setting. The next step was to work out how we would integrate that into that genome pathology setting. We've worked for a number of years now as scaling that up and integrating that within the VCCC Precision Oncology program initially, whereby our whole genome transcriptome work is really sitting here as an engine room. It partners very closely with the Pediatric Pathology and Stephen Fox, who's worked on comprehensive cancer panels. And then indeed under the guidance of Jayesh Desai and Stephen Lewin and colleagues within the VCCC, we've established a workflow whereby we're able to have a consenting process where challenging cancers are consented. They come in, there is a decision about what sort of test they should have. If there's not much found on a simple test, we may escalate. And then indeed, we're able to take that towards a molecular tumor board, the standard sort of processes we see for molecular testing right now across the world. I guess one thing that worked out for us in our favor early on is when trying to determine, the wisdom of Solomon as to what tests should you be using and when, and what should you be looking for. There actually was quite a lot of work done, coordinated by Stephen, to go cancer stream by cancer stream to get clinical consensus around an algorithm about what do you think are likely to be at this moment in time the best targets, the most obvious targets, the things we should be looking for in different cancer types, and how well does that match up to the molecular testing so that we could actually get the maximum utility from our studies. Finally, I think it's fair to say that the way we've settled at this moment in time is that we tend to be seeing that the cancer panels are being used on cancers that are well defined, cancers for which point mutations in coding sequence are known to be the dominant feature of that disease, where samples where FFPE is only available, our limiting material is only available, or if they are particularly low cellularity and need some sort of tumor cell enrichment. This is something that really we rely on the depth of the panel-based approaches to make sense of that. And then really what you're doing there is the primary goal here is trying to match the panel of coding mutations to targetable therapies which have that nice companion diagnostic approach already established. We tend to be using the whole genome sequencing in a different way. We focus on using it on patients that have ambiguity around their diagnosis, cancers that we know are particularly the dominant form of mutations we see in them may well be complicated, involving chromosomal rearrangements, creating of drivers. Hard to treat cancers is another area we've tended to prioritize because we want to get a very complete story quickly, and you may not have too many bites of the cherry when it comes, or time is working against you in some of those malignancies. And then indeed, we've been doing some work with Piers Bonbury and colleagues looking at how the same approach can be used for patients where prognosis is not clear. Clearly, with the wealth of both chemotherapy targeted transplant and indeed cellular therapies now possible, understanding prognosis in hematological malignancies is absolutely key. All right, so if we have a look at where we got to, I guess the last time we had a decent scorecard of where we got to the first phase of OCG, sorry, of the VCCC Precision Oncology program, where we're seeing that, and this really goes back to reviewing the outcomes of patients and indeed talk to the clinicians after the testing process to see how they were finding its use, finding that almost 2/3 of the cases we find clinically useful findings. And this is a combination of panel and whole genome, I should say. We're seeing targetable alterations in about 40% of the patients. We would estimate, well, at last estimate, 15% of the patients have their diagnosis refined or altered based on the molecular testing. And then finally, we've actually found that as this was being run forward, that the 50% of the clinicians found this to be really, really important in the decision-making process and thinking about future treatments of those patients. All right, so if that's where we were, we've been really trying to change gears in since the end of since 2020. There was a changing of gears, I should say, and that's really been possible through a strategic collaboration that was established between the University of Melbourne and Illumina, who would be known to any of you in the genomics space. This has been a really exciting partnership where we're looking to develop approaches to carry out scalable genomics, pioneer new opportunities in the applications of genomics to human health. And indeed, we announced in early February 2020 the Column Initiative. And this was a joint effort where we were looking to create a larger scale whole genome transcriptome pipeline for the sequencing of challenging cases. A challenging case, what is a challenging case? You can argue that almost any patient can end up being challenging at different times. In this case, you can see the inclusion criteria are quite broad, but we recognize that rare cancers where we quite often have no real, the foundational data about what drives those cancers is not nearly as strong as some of our more common cancers. We think that they warrant comprehensive testing. So in that case, our rare cancers were those that were 6 per 100,000 as a prevalence or less. We wanted to keep going with our analysis of recalcitrant cancers. So these are those that we know that standard chemotherapies have not worked well and they have a poor survival. We're interested in looking at relapse cancers. So those malignancies whereby they may have exhausted the standard of care and have acquired resistance to the preferred agents. And now we're trying to understand that mechanism and or looking for different targets. And then indeed, as you've heard before, if there are particularly enigmatic cases where we're not quite sure of the diagnosis, we want to see how we can improve those. And really our goals here are trying to create this platform to enable more of our cases to get on the clinical trials. We want to be able to improve those diagnoses. We want to see once again how we in a stepwise fashion can try and help particular studies cancer stream by cancer stream to take these sorts of testing on. And then finally, we're also very keen to test the overall, collect the information to show where the utility is in the comprehensive versus other based approaches. Just to highlight what we've been up to since kicking that off, you may notice that starting something in February of 2020 was almost exactly the same time as COVID hit. So despite those headwinds, we have been making steady progress when it comes to patient sequencing. And as you can see, we've tended to do this by partnering with cancer streams or clinical studies through which they already have frameworks, ethical frameworks, and the processes to consent and track their patients, and indeed have already got their own avenues for particular trials. And we've come in to try to see how we can enhance those studies. So for some of these we're effectively coming in, providing the engine and increasing the number of cases that can be processed under their banner. And that's an example of VCCC, the super programs that will look at cancer of the unknown primary, which have now got their own MRFF funding and have gone from strength to strength. Claire Scott's Stafford Fox Rare Cancer Program that we've been closely collaborating with for some years, and indeed looking at neuroendocrine tumours with Rod Hicks. There's a number of other studies whereby we have been, in this case, Colin is entirely focusing on a particular question. The registry really covers the most challenging cases that don't fit into some of these other efforts. There is a BALL study with Piers Blombury. There's a study I'll touch on related to having the pathologists initiate comprehensive testing rather than the oncologists. This is for diagnostically challenging cases. They may not be a cup, but it may not, it helps, we're trying to see how quickly we can remove ambiguity on difficult cases. And then indeed, there's a new study recently started off looking at mesothelioma, primarily using the whole genome to make sense of NF2 mutations, which is, there is now a trial going for the likes of NF2 as a particular druggable segment in mesothelioma. Many of those rearrangements can occur and be non-coding, so it's having the whole genome approach is useful for that. So you can see we're being quite pragmatic, and we're partnering with groups where we think we can get the best impact as quickly as possible. All right, so if that's really what we've been setting up, what I'd like to do now is spend probably 10 minutes describing to you how do we do what you see on this slide, and that is how do we go from a patient where we've sequenced their normal DNA and their tumor DNA, we've sequenced their tumor RNA, and we take the mass of somatic events, germline events that we see within that tumor on the left, and we're able to turn that into 4 or 5 statements that hopefully will be useful for that particular patient. And this is really what's been reviewed by the Molecular Tumor Board and is what is returned from our particular studies. All of what I'm going to show you now in terms of that engine, that pipeline, has been developed by Oliver Hoffman and his team. He's really been the architect of steadily refining this approach. We've worked through, I guess, over a decade now, or close to, the systematic moving from working in a traditional high-performance computing environment through now into moving to the cloud environment to carry out the sorts of studies that we're going to, I'll show you now. And I guess also what I want to highlight is when we do this, you'll see that there is a wealth of information that may have clinical and or diagnostic value within a genome. And we have got a number of modules by which we're able to, if you like, to summarize what we're going to get from that and pass that down the process, which will ultimately go into a report. So if we talk about the modules that we've sort of broken down cancer genome testing to, this is really the map of what that looks like now for DNA on the left, RNA on the right, and indeed when you put DNA and RNA results together in that 4th column from the left, and you can see that there's clearly a lot of insights that can be gained. I might just walk through some of these to give you examples of how we're using that information, and then you'll start to see later on the types of cases where those sorts of measurements are becoming useful when it comes to that particular case. So first and foremost, what we need to do is decode the germline sequences of that patient, identify all the somatic events that occur within that patient, and summarize the position of those on the genome and the nature of those events. If we want to understand all of those processes that can go wrong and the types of genomic events that are happening, you remember from the PCORG studies, we have to understand both coding and non-coding sequences. We have to understand gene copy number, chromosomal rearrangements, and point mutations. And suffice to say, there is now a near automated approach from when our samples finish sequencing, that they are moved off and this sort of primary analysis is carried out. This colorful diagram you'll see here is a Circos plot, which is just summarizing data from an individual whereby in one high resolution or high altitude image you can see here are the somatic mutations that were identified in B allele frequency along the outside, the actual location of where there have been point mutations in that genome, the relative copy number for those chromosomes, the presence of LOH within those particular regions as well, the structural rearrangements that we might see within those particular chromosomes, and indeed the blue lines going between translocations. And each wedge of this pie, if you like, corresponds to a chromosome. Chromosome 1, 2, 3, 4, going around, and the autosomes, and then the sex chromosomes up the top. So you can see here that these are now all being collated. They can be readily visualized. You'll see similar pictures like this going on when we get a snapshot of what we think might be going on in a patient's genome. All right, if that's the sort of global view, I think that there's a couple of ways we can look at this data. Some of it's simple, some of it's complicated. In a simple setting, if we're going through and looking at copy number variation, in many respects, all we want to know is what is the relative copy number of a particular gene, and that is a relatively simple amount of data to curate and review. But when it comes to structural rearrangements, it can get quite tricky that you've got these events. You're not just looking at the point of where an event has happened, you have to understand the orientation. In many cases, there may be multiple rearrangements, and it's very easy using a table to really not understand the true nature of the marker chromosome that may be within that patient. Standing on the shoulders of colleagues from the Hartwig, we do now have tools that are available that allow us to create, if you like, almost an NGS carrier type of chromosomes, which allows us to look at the context of that rearrangement and check 2 here, showing you exactly where the breakpoints were, that there are multiple copies of it, quite a simple event that we could look at that was occurring on chromosome 22. But actually, using these graphical approaches, we can now take some of these really complicated marker chromosomes, in this case, clearly created by a genomic catastrophe, because a multiple set of genes have ultimately come together. And indeed, not only can we detangle that mess, we can actually see and understand now that it is leading to the loss of a number of tumor suppressor genes, RB1, plus complete inactivation of P53 and the activin receptor. So these sorts of tools coming in together allows us different levels of altitude to resolve rearrangements now at a single nucleotide resolution. Another thing that's quite important to us, because we are doing a pan-cancer approach, working closely with Claire Scott, we do see quite a lot of gynecological cancers. We also see a reasonable number of head and neck tumors coming through VCC Precision Oncology. So understanding that the involvement of pathogens within that particular malignancy, identifying if there's viral integration, understanding if that viral integration has led to the amplification of oncogenes, or indeed the relative presence of the viral genome and whether that's active, of which there are many oncoproteins which they may express themselves, is something that we need to be able to do. And indeed, there are processes now to decode that, looking for the presence of viral sequences, and if that's triggered, carrying out these more detailed studies for all of our cases that go through the pipeline. Of course, we know crucially that tumor mutational burden is a key biomarker now in the treatment of cancers, particularly as it's seen as a biomarker for immunotherapies. And we're very keen that for any given cancer type, this is just showing you the plot of the relative frequencies that have been seen for TMBs across our reference set. When an individual turns up, in this case, it's the red line, we're able to actually have a look at where that patient sits. Clearly, this individual had an enormous tumor burden. So we're able to then include that within the report. Those mutational signatures I talked about before, we have a robust method for going through scanning the whole cancer genome for the presence of those mutational signatures, and then being able to quantify those. So this can be very useful in helping us understand. In this case, this is clearly coming from a patient that it's a skin-related tumor with a UV exposure leading to classical signature 7 damage. So that is a standard that we will use. One of the areas we think is really gaining a great deal of interest is the use of signatures and other features to make sense of homologous recombination deficiency. This is really coming about because we know that tumors that have profound deficiencies in the BRCA pathway are sensitive to PARP inhibition. It causes synthetic lethality, and indeed there's a wide number of cases where PARP inhibition is being used in BRCA mutated samples, whether it be ovarian, breast, prostate, and indeed stretching out to other cancers as well. One of the challenges we have with homologous recombination deficiency is it can be induced through epigenetic silencing rather than just mutations. And indeed, sometimes when we find variants within individuals, it's not easy to work out whether that mutation is likely to be pathogenic or not. But indeed, maybe, or certainly what we've seen from the likes of these signature approaches and other features, we can now confidently predict whether a tumor is HR deficient just using their genomic information. So these studies really come from Serena Nixanel's lab, and we've benefited from them greatly, been using them quite heavily now for some time. This is just the original data she showed looking at triple negative breast cancer, and the top is a rather colorful graph, but really what it's trying to show you is at the top is the relative presence of inactivating events within BRCA1, BRCA2, PALB2, RAD51C. The next block down is just summarizing the relative abundance of different mutational signatures, and we can see there is a preponderance of signature 3 in the BRCA deficient tumors, the bona fide ones. If you have a look at the rearrangement patterns of the chromosomes and the nature of those rearrangements, which we can do with whole genome, there's a clear delineation between the BRCA deficient and the ones that are BRCA proficient. And then finally, looking at microhomology and the nature of what happens to the sequences at that breakpoint. Once again, you have to have whole genome sequencing for that. You can see a clear delineation. It's not hard to then come up with a classifier that will define where you sit on either side of that curve. This was something that originally Serena and colleagues did in breast cancer, but indeed when they applied it to our Australian data, they were able to show that this had good predictive power both in high-grade serous ovarian cancer and indeed in pancreatic adenocarcinoma. And they've now gone on to show that this has really profound properties in pan cancer. Finally, they've actually used this approach to apply to FFPE material. And so that actually adds an extra string to the bow of this particular approach. So this is a routine measurement now we give on every single cancer that we sequence. We also use a second study that came out of the Hartwig Foundation's effort in the Netherlands. For those of you that don't know, this is a very large scale program sequencing metastatic cases. So the set they've used to create their classifier was a pan-cancer metastatic set. So we use that as well. We find very close concordance typically between these two tests, and has once again very good predictability as to pulling out the BRCA deficient cases from our cohorts. All right, so if that's really what we get from the global cohorts, there's a couple of other points about what else do we want to do. The orthogonal testing is great when we've got particular endpoints. We do end up with a massive number of events that may be hitting coding sequences. How do we actually quickly summarize those into the ones we think are likely to be impactful? And then how do we pull those together and put them into a report? That's really done through the use of a number of tools whereby the genes and the genome have been heavily annotated so that we know which ones are cancer related. And there have been large knowledge bases developed that hold mutations, the consequences of those mutations, and which ones of those may be significant clinically. And indeed, we have a number of tools like the Personal Cancer Genome Reporter, which was co-developed with colleagues to rank and prioritize the mutations that we see in genes into those that we think have a strong clinical significance through to those that we think are just a somatic change, may well be a passenger. And it's those tools that we use to sift down, take the absolute variants that we think are going to be the most impactful or the highest threat, and then we really drill down to those when going towards to make the report. The other thing I should highlight is we do RNA and DNA together. When that is carried out, really what we're looking to do is value add to our mutations. You can imagine if there's a potential gain of function mutation in the tumor DNA, no mutation in the normal DNA, we want to be able to confirm that that particular mutation is actively being expressed in the RNA to give us confidence that that is a driving event. So that's a very simple example of what we're doing with the point mutations. But we also know that there are a number of other variants that can be difficult to curate in a just with genomic information alone. A good example are splice site mutations. So if a mutation occurs close to the splice site in a gene, it's difficult sometimes there are in silico prediction tools to tell us whether that mutation is likely to be impactful, but indeed by combining both the RNA-seq and the genome results, we're able to identify places where there is clear exonic sequencing and when there are examples of clear, in this case, intron retention and indeed the expression of a coding mutation right at the splice site. So it's a great way to value add to the DNA mutations. Of course, RNA is also very useful in confirming the presence of oncogenic fusions if they're occurring. And indeed, you should also remember, as we've got all this information for that particular patient, we can quantify the relative abundance of that event. We can quantify the mutation, we can quantify the gene's expression. And that's really important where if we see an event, we think it's leading to an inactivation of the gene, we can now ask the question, does that particular event that we've seen have a concordant event within the RNA? And indeed what we do there is we compare the expression of that patient relative to the expression for that particular gene to what you'd see in a large cohort of matched cancer types. Finally, this rather crazy graph is to remind me that we also, when looking at regions of gain and loss, where here we've got a plot for an individual patient looking at copy number, and this is the relative expression compared to the TCGA cohort, we can look for genes that can have very high copy number, but indeed very low expression. We can look for genes that have very high copy number and very high expression. We can find regions that have been effectively inactivated when it comes to expression. Not surprising, once they're being deleted, we're seeing a loss of that expression. So a very valuable tool in picking the right oncogene in gains and losses. All right, so if that's, I guess, the summation of where our insights are coming from, if we have a look at some of the examples of what happens when we use that approach, I guess maybe I'll go through each one of those types of characteristics we're talking about, or inclusion criteria for the column study. The first is we want to be able to identify events within challenging cancers, and this is just showing a series of cases where patients have appeared for different reasons. They've been challenging. Some of these have been unusual, that they've lacked particular mutations. Some of them we know have an overall poor outcome. Some of these, when identified, were inoperable. And indeed, when going through and looking at these, you can see there's a wealth of different types of events that we've seen. And those particular genomic events we've seen have then ultimately given rise to the recommendation and been used as evidence to pursue agents to either compassionately or to get access on the trials based on the events that we see. Maybe if I drill down a little bit on one of those cases, example of a melanoma case that we recently reviewed, whereby a patient presented with a rapidly developing melanoma. They ultimately progressed on a number of different immunotherapies. The tumor had been sequenced using a panel-based approach and was shown to be mutation wild type. When we've carried out the sequencing of a metastatic biopsy, clearly we were able to show that it had the UV signature consistent with melanoma. The mutation rate was not overly dramatic compared to some melanomas. And when we started to have a look at the mutations of clinical significance similar to the panels, we weren't really seeing much. but in fact the details laid in some of the rearrangements that occurred within that tumour. Just showing you here, this is showing you actually a translocation that occurred in the patient taking out the far end of MAP3K8, and actually the translocation joined into a non-coding RNA sequence on the chromosome. And indeed, the RNA supported the presence of that 10-8 fusion and the generation of a effectively a truncated MAP3K8 open reading frame where the non-coding sequence again properly gave a stop codon. This was an interesting mutation to see, mainly because we knew from the literature just recently that when we look at the likes of melanoma, where typically we see mutations in BRAF, NRAS, and other parts of the MAP kinase pathway, in fact, MAP3K8 regulates part of that pathway and had been identified at low frequency in wild type or classical driver wild type melanoma tumors. And indeed, there's the study that came out at the end of 2019 has shown that if you have truncations or fusions that lose the terminal end of this open reading frame, you lose an autoinhibitory domain, and that leads to upregulation of MAP3K8. And indeed, they went on to show that in case studies where they've identified this, it was actually possible to treat these with MEK inhibitors. So what's, I guess, in showing here is that this is pretty much a cancer that's challenging because it's an outlier. The underlying root cause was a mutation that's atypical. We've been able to identify that quickly with that orthogonal data. We do a great deal of work in rare cancers. Rare cancers, we don't necessarily know what the playbook's going to be in those. We found a wealth of events in those ranging from novel fusions in known oncogenes through to homozygous deficient tumours that have then been sensitive to PARP inhibitors, and indeed other sorts of events that have been in very unusual tumours that we've then ultimately been able to get onto access of targeted drugs, or then indeed onto, in the case of mesothelioma, a good example of an NF2 event that then leads onto one of the local trials. In terms of the RET fusion, this work was recently published, and this time I'll just skip over it. This is just showing you a good example of how in a pheochromocytoma, these are very rare adrenal tumours, not typically known to be driven by RET fusions, but what we're seeing here is the combination of a novel gene with that RET, but effectively creating the oncogenic REC open reading frame that can be readily expressed in that tumour, and indeed in this particular case, showing a rather exceptional response to selpercatinib. Once again, being able to be detected very quickly through the combined whole genome transcriptome approach. I guess just in closing, I should say that what we've seen for this issue of drug resistance, for many of the cases where they've run through the gamut of standard therapies, there are a number of events that we can readily see within the genome and or transcriptome. This is just highlighting some of those events that we can see here. Certainly acquired immunotherapy resistance, finding those events that are happening in antigen presentation is readily identified in the DNA. A number of other ways to avoid PARP inhibition in some of the BRCA deficient tumours as well is something that we've seen. And in some cases, we've seen the acquisition of more aggressive oncogenes in those tumours, which them in themselves are readily targetable. I think the Diagnostic Conundrums is something that's an early project, but we're finding a very high yield of these. We're actually finding that, and I think this is really a credit of Catherine Mitchell and Owen Trial, that they're using these approaches judiciously to tackle really challenging cases where there's a number of differentials, and the molecular data can come in and really help break the tie in some of these really unusual cancer types or subtypes of rare cancers themselves. Just then in closing, I think that I think you can see there we've got a relatively robust approach for steadily now working through whole genome transcriptome in cases. We do recognise, however, that sometimes that's not going to be enough. This is an example of a case we recently had of a uveal melanoma. We fully recognise what the mutations were that were driving the disease. The mutation profile is giving some very clear indication that it's a poor prognostic type. Unfortunately, there was no therapies available for that repertoire of events within that particular patient. However, what we've known is from the recent advances in literature is there are a number of immunotherapies that are steadily being tried for uveal melanoma, in this case, for those that are expressing a particular type of deranged CD8 T cell. And indeed, what we've looked to do is start to bring some of our other new research tools like single cell analysis and integrate that with our diagnostic pipe as a way to start to get rapid readouts for these particular cases. And in this instance, what we see in the reports is that there is a co-expression of inhibitory molecules LAG3 and PD-1, and indeed there are now approaches that can target that LAG3 involvement. And for this particular patient who's taking we're lucky enough to rapidly get fresh material and carry out single cell analysis, we were able to show high level of co-expression of LAG3, which was then used as evidence to get access to drug for this particular case. Finally, I think we all know that cancer testing is now moving towards the likes of looking at liquid biopsies rather than just tumours tissue alone. And we're very keen to be riding that wave. We've, in the first instance, doing a lot of work now using a comprehensive panel for that. So starting with the first step, that lowest rung, and using that to complement the tissue-based studies that are carried out by Stephen Fox and co. I think we've really stepped into that first and foremost because the depth of sequencing that is needed for this assay is enormous. It is an assay that is potentially very rapid turnaround. It relies on cloud computing. It'll actually give us mutation readouts on coding sequences of 535 genes, and it has the potential of identifying common driver rearrangements in a key set of genes, and indeed can identify large amplifications of key oncogenes. And this is an assay that we're readily working up in the lab to then see how we can integrate that with the other repertoire of studies that we've got. We're currently in an observational mode, but we do look forward to opening that up to more studies in 2022. So finally, I think I'd say that all of this really stands on the shoulders of this entire team that comes together around this workflow. I think we're in a process now of up-gearing that and adding new assays, trying to work out how do we scale up the reporting process. There are some cases that are simple. There are some that are truly worthy of the molecular tumor board we have now. And I think that 2022 will see us trying to speed up that process. All right, just in closing, the work I'm presenting really comes from an enormous team of people. I need to acknowledge Oliver Hoffman. He's the co-PI on the activities we have with Illumina. He leads the informatic efforts and really has been the one responsible for building our pipeline. I should also acknowledge Kim, who is the manager of our clinical genomics platform, and Yoop, who is leading our curation team for all of our cases going forward. There's a large number of people across this city. I should be acknowledging colleagues across Australia, our funders, and indeed my colleagues from the ICGC. Thank you.
Great, thanks so much, Sean. It's a real tour de force of the cutting edge of where this field is at. We've actually we're a bit over time, but there's so many questions popping up. I think we have to at least honour some of them. So I'll just cut to them straight away. So the first one is from Baumik Shah, medical oncologist. Do we have a mutation signature for asbestos? No, we don't. There has been an argument that there is a signature that is asbestos related, and there's no doubt that there is a signature 40, that is seen and is quite common in mesotheliomas, but we also see it in other malignancies as the dominant signature. So not nearly as, there's an enrichment, but not as specific as we'd like it at this moment in time. Next question is from Nick Wong. Yeah, so could you comment on the relative contributions and needs ongoingly for computational tools versus at least additional human interpretation around and variant curation and interpretation. So how much of this whole process is becoming automatized through?
Yeah, great question. It's a great question, Nick. And really where we are right now is we are still very much in that process of relying on human curation for, we can get to a particular point where we can stratify, and it's very easy to get the things that are strongest, clinical, the obvious things are very easy to pick. What we find though, that, and if you have a look, I think when you look at challenging cases, they do need extra curation. I don't think we have the big data yet that we need to be able to create the tools to do this in a truly automated fashion, but they're coming. We're seeing the data sets being generated by Genomics England, Hartwig, other national activities. All patients now in Japan have access to molecular testing, at least comprehensive panel, and then going up to whole genome and others. That's where the data will come from those millions of cases. It's coming. It won't be here this year, but I think that next 2 to 3 years we'll see huge changes.
Finally, from Vicky Marshall, what's the how reliable are the sort of panel approaches for inferring mutational signatures? Yes.
So mutational signatures, I think if you just saw the pattern of those crazy scars, some of them are really distinct spikes. So they're going to be very specific nucleotides that get damaged. Others, they're a far more complicated pattern. There can be waves and there can be crests. Depending on the complexity of the signature, you need more mutations to be able to call it accurately. So that means things like UV signature, which is those thymine dimers being mutated by the UV. That mechanism is well understood. It's very specific, and you can call those quite well on panels. But the likes of HR deficiency and many of the other signatures, you're going to struggle. And the lower the cellularity of your sample, the more you're going to struggle again. So, for instance, for us, we need 1,000 mutations, somatic mutations, for us to be absolutely certain about the signatures. You're never going to get anywhere near that, even in an exome.
Mm.
Okay, thank you.
And one very final one from me. With respect to use of sequencing to help us identify the tissue of origin of a given cancer, can you comment about the relative usefulness of just direct DNA sequencing versus bisulfite sequencing to look at DNA methylation?
Yes, so the graph that I showed you whereby PCORG was looking at, we can determine the tissue of origin of a tumor just looking at the background passenger mutation rate, is effectively a proxy of bisulfite sequencing. So we know that if we just look at the mutation rate across the genome in 1 megabase bins and you walk through it, there will be a pattern that corresponds to chromatin occupancy. And we know that regions of active transcription have transcriptional mediated DNA repair, so their pattern of damage is different. So in fact, we can now very accurately use that readout to come up and make sense of what's going on. Now, if you think about all of the other information I've described to you, from signatures to background patterns to rearrangements to all of that, and you take all of the data from PCORG, you can come up with a set of 7 features that are just present in tumours to call tissue of origin far more accurately than we would by some of the other approaches we've done. Richard Tothill, who is a member of the centre here, is very actively pursuing this and has been working closely with colleagues in the Netherlands, looking at both DNA and RNA put together here to call tissue of origin. And maybe you should get him over to give a chat here sometime and he could wow you.
Fantastic. Thanks very much, Sean. It's a really amazing talk, and I've actually got a pretty good turnout, and many people have stayed on to the end, which I think reflects not only your standing generally, but the quality of what you've been talking about. I also actually want to just give a personal shout out as well. So Sean, probably not known to many people, has actually really been he's really been very helpful to our own program with respect to attendance at and really some very meaningful contributions to our own developing molecular tumor board series. You know, he's done that in his own time and, you know, just out of interest. And in fact, he's actually taken on a handful of cases where the results of his extended profiling have been fed back to those tumor boards and actually influence the management of individual patients. So it's really a fantastic contribution. We look forward to ongoing and increasing collaborations going forward. Sean, we've just got our own program refunded for the next 4 years, which we're really excited about, and thinking about all sorts of ways to take that to the next level. And I think one of the opportunities for us is to work out ways to better integrate with the program across the river that you oversee.
Fantastic, thanks, Mark. Congratulations on the funding. It's always a pleasure to present this. I'm standing on the shoulders of a lot of people, and I guess as we come out of COVID, that opportunity there is really to see how we can do more together going forward.
Thanks. So thanks everyone for turning up on a Friday when you really want to be going out there and hitting the shops that are soon to be open. It's a big commitment to stay on and listen to the talk rather than preparing for that. So much appreciated. Our next seminar, Anna, if you could just pop up the slide. So our next seminar is on the second Friday in December, the 9th, by one of our leading academic surgeons in the precinct, James Lee. He'll be talking specifically about his work on the genotyping of thyroid cancer. So please put that in your diary. We look forward to seeing you there. You can keep generally up to date with our upcoming seminars via the MPCCC website at www.monashpartnersccc.org. Thanks very much again, Sean. Thanks everyone for attending, and have a great weekend and enjoy burning the credit card. See ya.
Thanks.